So I'm going to talk to you a bit today about cervical cancer screening and HPV testing in the era of the HPV vaccine. Now, just an overview of the lecture. So I'm going to talk to you about the epidemiology and molecular biology of HPV and cervical cancer control, screening in the era of the HPV vaccine, and the role of HPV testing currently and in the future. So why are we talking about HPV? Well, we're talking about HPV because it turns out that HPV is central to the development of many genital tract cancers, and most importantly, cervical cancer. And although we've essentially wiped out cervical cancer in the United States through 60 years of Pap screening, it remains the second most common cancer in the world, especially among women in the developing world. HPV is also responsible or central to the development of vulvar, vaginal squamous cell cancers, penile cancers, some squamous cell cancers of the head and neck, and possibly plays a role in squamous cell cancer of the skin, although this is, no one's quite sure about this. The molecular structure of HPV, I'm just going to spend a second on this. It's a double-stranded DNA genome, and the capsids, the surface of the virus contains the L1 and L2 proteins. The L1 is the major capsid protein, is important because it's highly immunogenic, and this is the epitope that is important in developing neutralizing antibodies. And the L2 is a minor capsid protein, is less immunogenic, but has more cross-reactivity, and as we're going to see, it's also probably important. So just briefly, the biology of HPV. The most important thing about HPV is you can't culture it. And this has really a lot of implications for the study of the virus. Well, first of all, it also means that for classification is done by sequence homology. Further, detection of the virus has to be by hybridization assays, since there's obviously no way to grow it. And I know you guys are all interested in tests, so I'm just going to talk for a second about the fact that there are a variety of sensitive and specific assays that are now commercially available. And the kit assays include the Roche line blot assay. This is a research-only assay, but it's the gold standard essentially for large epidemiologic studies. What you do is you use a L1 consensus primer and then take the PCR products, which are typed using a reverse line blot assay. Highly sensitive and specific, but it's very time-consuming and costly, so it's not appropriate for clinical use, but great for research-based studies. The most widely used assay, which used to be called the Gene Assay, is now Kaijin, and this is an FDA-approved assay for HPV-based triage of ASCUS. What that means is that women who get a ASCUS or an atypical diagnosis on Pap screening are now tested for HPV, because we know that cytopathologists cannot distinguish between things that are atypia that's real and atypia that's having a bad day. And so by using the HPV testing, we know it depends on, we decide who's going to be sent for colposcopy and biopsy. It's a extremely widely used test. It has some great points and some drawbacks, including the fact that there are no internal controls that are supplied with the kit. So you actually never know if the test is negative, or you just don't have enough sample. There's a new FDA-approved assay out just this March, a hologic HPV genotyping assay, very similar to the Gene, but it does have controls. They all have plus and minuses. Now, then we just to go over the most widely used test, this is the Kaijin or the Kaijin test, and essentially what you do in this test is it's done out of the pap smear material. You denature the exfoliated cells, hybridize with HPV RNA probes. Then you capture the hybrids with monoclonal antibodies on microtiter plates. You use a second a conjugated alpha phosphatase conjugated monoclonal antibody, and then you have your alpha phosphatase splits a chemiluminescent substrate to produce light. It's fast and it's highly sensitive. We've also, in our lab, we've developed a variety of HPV tests, and the latest one we've developed is a Luminex-based test for HPV typing. The Kaijin test is not type-specific, and I've given you the reference if you're interested in looking at it. Now, what I didn't talk about in this when I went through the test is any serologic assays, and the reason is because from a clinical standpoint, I was thinking today, are there any possible uses for a serologic assay as far as HPV goes? And the only thing I could think of is, I guess that if you had the right assay, you could possibly test people to see if they had been exposed to 1618 or one of the ones that are included in the vaccine to see if they wanted to be vaccinated. However, it's not practical because of the fact that, well, first of all, the serologic tests we have are not particularly sensitive. And the other problem is that the vaccines, it appears that although, for example, the GSK vaccine is made for 16 and 18, there's a lot of cross-protection. So that the antibodies that are raised against 16, 18 appear to provide cross-protection for some other types. So looking at the antibodies is really not going to tell you very much about who needs to be vaccinated or not. Okay, so now that we got the tests all the way, the big question in HPV is not being able to detect the virus because, as I said, there are lots of good ways of detecting it now. The real question is we're going to see is what is the role of HPV testing in cervical cancer control, and that's what I'm going to be talking to you about today. But first, obviously, the fact that one last point about the fact that you can't culture the virus, obviously this was the limiting factor in developing the vaccine because we didn't have any antigen. There was no source of antigen. And so it had to wait until we could, cloning and all. And at that point, after that, everybody knew that the vaccine could be readily made. So the classification of HPVs, just let me tell you that they're divided into types, subtypes, and variants on the basis of genome homology. And the types and subtypes and variants tend to differ by the tissue most likely to be infected. For example, HPV-1 only infects the soles of your feet to give you plantar warts. Doesn't infect any other kind of epithelium. HPV-4 only infects the trunk, for example. So they're pretty specific. Mode of transmission, obviously there are ones that are sexually transmitted and others that are non-sexually transmitted. And the biologic behavior differs. Some of them are high-risk types that are associated with cancer, and others don't have any relationship to cancer. So in common parlance, people talk about high or low risk types of HPV depending on the strength of the association with malignancy. And they talk about cutaneous or mucosal types. The mucosal types are the ones that are sexually transmitted. And, or they can talk about skin versus genital types. Okay, so there are over 100 types of HPV, and believe me, we're not going to talk about them all today. The only thing I'm going to tell you is that there are about 30 sexually transmitted types. 15 of those are high-risk types. There are also 70 cutaneous HPVs. The most commonly discussed low-risk types are 6 and 11. You'll hear about those because they're the cause of vulvar warts. And the most common genital high-risk types are the 16-18s and the 30s and 45s. And you hear about those because 16-18 is of particular interest because together those account for between 60 and 75% of all cancers. And there's also some data, including our own data, that shows that the malignant potential, the oncogenic potential of 16-18 may be greater than all the other high-risk types. Okay, so we've done that. Okay, so what are the clinical manifestations of HPV infection? Well, this is clearly, and I'd like to know if anyone else has any ideas. I think this is, HPVs are the most common viruses. They are the cause of, for example, of all skin, of all cutaneous warts, and essentially by all children are essentially 100% are seropositive. I said 95%, but the literature says it's even more. So essentially, we all acquire HPV infections very early in life. They're also the cause of the most common sexually transmitted diseases. So they cause genital warts. They're also responsible for flat warts, which are otherwise known as cervical intraepithelial neoplasia on the cervix, and are essentially responsible for all the abnormalities present on pap smear. When we look at pap smears, that's essentially what we're looking for, HPV-related changes. That's it. Certain types of HPV are also central to the pathogenesis of many cancers. Okay, so what are the statistics? Well, in women who are 25 or less, the prevalence is somewhere between 28 and 46%, and that's for high-risk types. By age 50, 80% of the population of women have acquired HPV infections. And so the estimated prevalence is about 20 million. The height of positivity is between 16 and 24, that is, with the onset of sexual activity. And we're talking here about genital HPV types, which are really what our focus is on. Okay, so transmission and epidemiology of these genital types. Well, transmission requires direct contact with stratified, squamous, metaplastic, or columnar epithelium. The presence of the genital types, unlike other STDs, there's a very high rate of transmission with a single contact. However, the prevalence of differing genital types does increase with the number of sexual partners. And as I said, the peak prevalence of incident infection is associated with the onset of sexual activity. The prevalence also appears to increase in older women, and this is thought to be due to decreased immune surveillance. So it's believed that once you acquire some of these high-risk types, you never get rid of them. For example, in our studies of HIV-infected men who were celibate for many years, as the CD4 counts decreased, the number and the prevalence of HPVs increased. So I'd like to go over how high-risk HPVs cause cancer. So in order to do that, we want to talk a little about the organization of the HPV genome. And essentially, this is the simplified version. There are three important regions. There's the regulatory region that controls transcription, and this contains binding sites for transcriptional repressors and activators. Then there is the early region, and they call it early because these are the genes that are turned on first, and it contains the genes. The HPV genome only has 8 genes, and 6 of them are in the early region, and they're responsible for viral replication. And importantly, this region contains the 2 HPV oncoproteins, which are E6 and E7. The late region contains the coding for the structural capsid proteins L1 and L2. So what do these oncogenes do? Well, most high-risk HPV infections are self-limited and without consequence. And only the high-risk types contain these or produce these oncogenes, E6 and E7. In the self-limited high-risk infections, we think that the infection starts in the basal cells, and the virus resides outside of the host DNA. It doesn't integrate. It's episomal, and there's a very low rate of viral replication. And if you look at the level of DNA, the copies of DNA, it's very low. And likewise, the level of the E6, E7 oncoproteins is very low. If you move up into the suprabasal cells, so now these cells have divided and their daughter cells are in the upper layers, the HPV, every time the cell divides, also divides. And what you see here is vegetative replication of the viral DNA. You also start seeing expression of the capsid proteins to form viral particles. And so you get assembly of the virus. And then as the cells move up in the stratified squamous epithelium, as in the terminally differentiated cells in the stratified squamous epithelium, the cells essentially die and fall off. And when these cells die and fall off, they release their HPVs. So it's a very tricky virus because now it sort of can evade the host immune response. First of all, it's completely intraepithelial, so it never causes viremia, and so many parts of the immune response of the immune system don't ever see it. The other thing is there's no cytopathic effect. There's no abnormal cell death. It just is released when the normal epithelial cells fall off, and so there's really no inflammatory cytokine response. And then lastly, the high-risk E6, E7 proteins inhibit the innate immune response and they block the functions of IFN. Now, despite the fact that it can evade the immune response, most high-risk HPV infections are resolved by the immune response within a year. So what you see serologically is development of the antibody response from about 1 to 2 years after you see the initial detection of the HPV DNA. However, some women are unable to clear high-risk HPV infections. And in this case, they develop persistent HPV infection. And we and others have looked at why this is true. It's partially immunologic, partially the exposure to other factors that promote cell replication. But in any case, in some women, you get the establishment of persistent infection. Now, persistent infection, of course, means that there is continued production of the E6, E7 oncogenes and continued exposure of the host to these oncogenes. So what are these? And this, of course, is associated statistically with an increased risk of carcinoma in situ. So what do these oncogenes actually do? Well, these oncogenes inactivate the host tumor suppressor functions, which are related to control of the cell cycle, and they also counteract apoptosis. And specifically, E6 binds and inactivates wild-type p53. It also upregulates telomerase so that the cells don't die, and the epithelium becomes thicker, and the virus can keep reproducing. E7 inactivates RB. So what happens is essentially that you're setting the stage for cancer. You're essentially the fact that it inactivates p53 means that if you're exposed to other carcinogens, that you can't repair the DNA. There are studies that suggest that HPV itself can increase the rate of mutation and epigenetic changes. But there's also a lot of data suggesting that HPV is necessary but not sufficient for development of malignancy. And in fact, you have to have subsequent exposure to other carcinogens, like, for example, infection, a lot of inflammation, HIV infection. The strongest cofactor for cervical cancer after you adjust for HPV infection is actually C. trichomonas infection. And it's thought that this is because it causes a huge inflammatory response. And also it may cause epigenetic changes, since it is a obligate intracellular parasite. So we've gone through what the self-limited infection with high-risk HPVs looks like, and we've gone through what persistent HPV infection looks like. So what does cancer look like? What is the HPV doing in cancers? Well, unlike the precursor lesions where the HPV genome was always episomal, when you look at cancers, you see that fragments of the HPV genome are integrated into the host chromosomes. Now, the mechanism and timing of this is really unclear. So it's not exactly clear if this is causing malignancy or if this is a consequence of malignancy. But very few precursor lesions, with the exception of carcinoma in situ, contain integrated HPV DNA. Now, there are no integration hotspots, for example, like there are in other cancers, although the integration does occur more often at fragile sites. However, what is important is that the integration of the HPV genome into the host always disrupts the E2 HPV genome, which is which regulates replication. So essentially, you have disrupted the normal control of viral replication. When you look at the integrated fragment of viral DNA, you also find that it also contains the E6 and E7 genes, and that it's transcriptional promoter, which means that once this stuff is integrated, you have unregulated replication of the E6 and E7 protein. So, the last thing I want to say, the point I want to make about integration, is that if you look at cancer cells, you now have integrated HPV, but you could also have episomal HPV in these cancer cells. And there's some evidence that suggests that the E1 and E2 proteins, which are produced by the episomal HPV, contribute to or increase the risk of mutations because they lead to the replication of the integrated HPV and the flanking cellular DNA. So this is one way, for example, that HPV might cause mutations and epigenetic changes without exposure to other co-carcinogens. Okay, so this is just a visual thing that tells you that in the precursor lesions we see episomal HPV, while when we see it in the cancers we see integration, and we have disrupted the E2 area, and what we find is that it always contains the E6, E7 genes. Okay, so to summarize the pathogenesis of HPV and of cancer, of cervical cancer, what we see is HPV infection, and probably partly because of immunologic factors, some women are unable to clear the infection. We get development of persistent HPV infection. And then with exposure to other risk factors, carcinogens, we get cellular deregulation and then progression to the malignant phenotype. So to summarize what I've said so far is that HPV is necessary but not sufficient for progression to cancer, although we've seen there may be mechanisms by which it also itself can cause mutations and epigenetic changes. But it's thought that HPV sets the stage for the accumulation of chromosomal defects that allow the development of the malignant phenotype by its binding and degrading wild-type p53 MRB and upregulation of telomerase. Okay, so as I'm sure you're all aware, recently there have been 2 vaccines that are against prophylactic vaccines against HPV 16 and 18, which, as I told you, are the main causes of cervical cancer. And these vaccines, I'm not going to spend a lot of time talking to you about them, except to tell you that they are amazing vaccines. They're highly effective. And as I mentioned earlier, everybody knew that an effective vaccine could probably be developed because, for 25 years ago, there was an effective vaccine developed for canine papillomavirus infection. And of course you could collect lots of warts from laboratory animals to make the vaccine. So it was easily made. The problem with the development of the human papillomavirus vaccine was that you need conformationally correct epitopes of the L1 protein, which, as I told you, contain the neutralizing epitopes, in order to assure induction of neutralizing antibodies against the HPV. And since the virus couldn't be cultured, there was no way that one could get adequate amounts of antigen. So with the development of expression systems, then the vaccine could be easily developed. So it's a highly effective vaccine. It's probably it requires 3 doses now, but there are a number of studies showing now that suggest that we won't need that many doses. And we just finished a randomized clinical trial in Africa where we vaccinated 9 and 10-year-olds, and it looks like it really will be the solution to cervical cancer, development of cervical cancer in the developing world. So what will be the impact of the HPV vaccines on cervical cancer control? Well, we're not sure. Clearly, factors that will determine its impact include the number of HPV types that are included in any vaccine. Although it's clear now from the GSK data that although the vaccine was made to target or protect against 16-18, there is a lot of cross protection to other types. So it may not be everyone was worried about, how many types can you protect against in one vaccine, but it may be an easier problem than we all thought previously. The efficacy of the vaccine will depend on whether the efficacy for not only the types but the variants. And there's very little data on this at this time. Cross protection I discussed. Duration of protection. Well, the first this vaccine has only been around for about 6-7 years, so far so good. But the antibodies titers that are induced by these vaccines suggest that you will have long-term protection. And then, of course, the question is what % of the population is going to be vaccinated? And we all know what happened with hepatitis B and how long it took to get a significant proportion of the population vaccinated. And then the last thing I put up here is the behavior of other high-risk types of HPV that are not included. I mean, it's theoretical possible that when you protect against 16 and 18, that you'll get a lot of cancers that are related to the 30s or the 50s or something like that. However, as I mentioned earlier, 16 and 18 appear to be more oncogenic than the other types. So I sort of doubt that this will happen. That's what our data says, but we'll wait and see. Okay, so now that we've got a vaccine, do we need to still do pap smears? Do we need to continue with cervical cancer screening? And unfortunately, we are going to need to continue for a while. And why? Well, first of all, the current vaccines do not protect against all high-risk HPV types. It could be that there is a lot of cross-protection, at least with the GSK vaccine, which has a different adjuvant than does the Merck vaccine, but we don't know. Okay, but most importantly, these vaccines, these are prophylactic vaccines and don't protect against anyone who was already infected with the HPV virus. And so that means that anyone who is essentially over 14 years old is not going to be fully protected by these vaccines. So what it means is that screening for and treatment of women with relevant cervical cancer precursor lesions will at least need to go on for the next 50 years. And of course, if we don't have widespread vaccination, then we'll need it for even longer. So now the question is do we need to change our screening approach, and what's going to be the role of HPV testing in cervical cancer screening now and in the future? Okay, so in order to answer this question, I want to just review with you what we did for cervical cancer control prior to learning about HPV and its importance in cervical cancer. Well, cervical cancer control has always been based on the concept that invasive cervical cancer arises from precursor intraepithelial lesions which can be readily treated to prevent progression to invasive disease. Now, classification of these precursor lesions is currently based on what's called the cervical intraepithelial neoplasia system. Now, this system was developed about 40 years ago, obviously prior to our understanding of HPV infection. And I'm sure that most people have seen these little diagrams that shows an epithelium, and it shows the progression of CIN1 goes to CIN2, goes to CIN3, and then it goes to invasive cancer. And it turns out that this is just wrong, but the idea was that these things are consecutive, progressive, non-reversible stages in the development of invasive cervical cancer, and this led to the belief that most CIN1s would likely progress to CIN3 and eventually invasive cancer if they were untreated. And because of this, there were about 50 years where we essentially treated all CIN lesions by ablation, by LEAP. And we did this, so what we did is we had all women go for annual cytologic screening, and we knew that a single pap smear had a very low sensitivity for detecting underlying CIN lesions, only about 30 to 50%. And thus, to increase the sensitivity, anybody with any kind of abnormality on a pap smear was sent for colposcopy and biopsy. And then anybody with CIN or even ascus, repeated ascus, then underwent LEAP. And this was highly successful, and you can see that the cervical cancer trends in the U.S. Went down markedly as we essentially got everybody into pap screening and treated all these cervical abnormalities by LEAP. But essentially what we were doing, it turns out, was that we were treating warts. When you look back, it really didn't make a lot of sense because the natural history studies that were around even before we knew about HPV infection showed us that less than 1% of CIN1s or ascus, if left untreated, would ever actually go on to invasive cancer. And likewise, CIN2s untreated, less than 12% would go. And in fact, if you leave carcinoma in situ untreated, only about 30% would ever go on to invasive cancer. The problem is the pathologists don't know which 30% it is. So obviously you have to treat all of the carcinomas in situ to prevent the 30% of those women who have it that would go on to get invasive cancer. But essentially, what we have done in the past is that we have decreased the incidence of invasive cervical cancer by surgical ablation of essentially most HPV-related lesions. Now, this was great, except for we figured out that that there's some problems with it. So after HPV was discovered and we began looking at the natural history studies examining the development and the natural history of CIN in relationship to HPV infection, it became very clear that CIN lesions were just the morphologic manifestation of HPV infection. And you got an HPV infection, and several months later you developed a CIN1. But in fact, about 90%, over 90% of these lesions would resolve themselves within a year. We also figured out that many CIN 2 to 3 lesions really have nothing to do with cancer, especially those in young women, but they are an immediate consequence of HPV infection, and many of those appear to have a high rate of regression. But most importantly, what has come out is it turns out that ablative treatment of CIN by LEAP is associated with subsequent obstetrical morbidity. So now, what we thought was a very benign procedure, LEAP, turns out to be not as benign as we thought. So the new model of the pathogenesis of cervical cancer in relationship to HPV infection is that HPV infection, the immediate consequence of HPV infection could be CIN1 and CIN2 lesions. You get a persistent infection. Some of those look like they look like they're high-grade lesions, but the only really precursor lesion that we know has a high risk of progression is probably carcinoma in situ. The problem is that morphologically we can't differentiate the lesions that will go away from those that will go on, the high-grade lesions. So obviously what we need here is molecular markers that will help us do that. So now the question becomes, now that we know all this and we know that we've been over-treating many lesions, but we're still going to we can't vaccinate everyone because a lot of us have been infected already. The question is how to best identify women at risk for development of ICC. And so given the new understanding of the natural history of CIN, and the other problem is going to be now that we have this population that increasingly will be vaccinated. And this is going to complicate things because the prevalence of the disease we're looking at is going to decrease markedly. Okay, so how can we do it? Okay, well, first I want to tell you what the current recommendations are for screening and management of cervical lesions. And this has become increasingly complicated because little by little, what we learned in the research area is being moved, this is truly translational research, into the clinical practice. But it's a little confusing at this time, and you'll see why. Okay, so first of all, there are many different recommendations that are issued by the different groups. The ACOG has one, and CAP has another, and this one is from the Allied Task Force, and they're not always in line with each other, and it's actually very confusing to primary care docs. So this latest recommendation suggests that cytology screening should only start at age 21, and this acknowledges the fact that what we've been calling dysplasia, or CIN, prior to age 21 is probably just self-limited HPV infection. We don't want to know anything about it, so let's start at age 21. Women it's very rare that women 21 or less develop cancer. The next thing is that we're going to screen every 2 years, and this will help us avoid detecting HPV infections, because hopefully if you get one, you're going to get over it and you're going to have a negative pap smear by the time you get screened again. Women who have ascus, which is atypical squamous cells of uncertain significance, those women will undergo HPV testing, and only those who are HPV are positive for high-risk HPV types will be sent for colposcopy and biopsy. All those with a CIN lesion will be sent for biopsy. In women 30 and older, what these people suggest you do is that women be screened by both cytology and HPV infection. This may not make a lot of sense, because they say that if you have an abnormal pap, or if you're HPV 16 or 18 positive, then you should go for a biopsy. So if you have an abnormal pap and you're negative for all high-risk types, you still go for a biopsy. So I'm not sure that makes a lot of sense, but that's what the current suggestion is. If, however, women over 30 are positive for another high-risk HPV type, then they don't have to be sent for colposcopy and biopsy, because those types, as I told you, are supposed to be less oncogenic, but they have to come back in a year for another HPV another HPV test. Furthermore, on biopsy, if it turns out that you have CIN1, that's okay. We're going to follow you. It'll go. Most cases, it'll go away, and we're only going to treat now biopsy-confirmed CIN3. So both the screening recommendations and the management of cervical lesion has markedly changed over the last 10 years as we've gained more insight, as I said, into the relationship between HPV and these lesions, which we used to think were all precancerous lesions. So now it would seem logical that if you're going to screen, you're going to increase the efficacy of your screening, that you would somehow incorporate HPV screening, which if you don't have HPV, you're not going to have cervical cancer. So there's a lot of work going on now to try and figure out how we're going to use HPV screening, how we're going to screen in the future. Okay, so the current recommendation, which is the first one, is that you do primary screening by cytology with HPV testing for triage to biopsy of ascus. So remember, about 15% of pap smears have the diagnosis of ascus. That's millions of women every year get that diagnosis. These women used to be sent for colpo biopsy until we figured out that most of them don't have, are really not at increased risk. And so what we do now is we test for HPV, at high-risk HPV. If they're negative, we figure they're not at any risk, and if they're positive, we'll biopsy them. The second possible scenario is that you screen on the basis of HPV detection. So you screen for high-risk HPV, and then you refer all those who are positive for high-risk HPVs to colposcopy and biopsy. The third possibility is that you screen by using HPV, and all those who are positive you do a pap smear on. And the last one, which I think is the only logical one, is to screen on the basis, for high-risk HPV, and then use another test that shows you whether among the people with HPV you've developed any molecular changes that actually suggest that you might be at risk for cancer. In other words, you're moving down the pathway that is associated, that you have changes that are associated with cancer. Okay, so let's just talk about the shortcomings of the currently recommended approach. Well, the first thing is, as I told you, cytology only has a sensitivity for CIN3 of about 30 to 50%, and for a single smear. And the other thing is that cytopathologists are not reproducible in their diagnosis. And it really depends on what case you saw before and after, what you're going to call this case. The next thing is, and I just want to point out that there are screening machines, but they don't help either, because all they do is they screen a population and give you 50% to look at them again. So you still have it's still very subjective. The other thing is that because cytology is so insensitive, remember we're sending these people with ascus to colpal biopsy. Now HPV triage does reduce the number of people we're sending to colpal biopsy, but remember, for women 21 to 29, these women are at high risk of having sequential Hpv infections, all self-limited as they get new partners. So you've got a really good chance if you're between 21 and 29 of having a positive Hpv test and being sent for colpal biopsy. Very expensive. Okay, so just to show you, we actually did a study of 4,000 women to look at all these different approaches. And this just shows you that for women less than 30 years old, the current approach, that is cytology with HPV-based triage, has a sensitivity of, this is for CIN3, of about 50%. It's highly specific, 90%, that's good. You refer less than 30 years old, you're still going to refer 14% of the population, and that's a huge amount since you figure you're screening all the women at this age. If for women over 30, the sensitivity is not very good specificity, and of course you only refer 7%, and that's because the over 30, you're much less likely to be getting these HPV infections because you've already been infected and you don't get them again. Okay, so the problem is that this does not overcome the innate insensitivity of a pap smear, and as I said, young sexually active women consecutively acquire and resolve different high-risk HPV infections. Okay, so what about the second scenario? Primary screening based on detection of high-risk HPV types, and then you just refer everyone for colpal biopsy. Well, if you look at the women who are less than age 30, it's got great sensitivity, 90%. But your specificity, you're going to refer 33% of your population of all women less than this to colpal biopsy, which is clearly impossible. And so your positive predictive value is very low. Your negative predictive value is high. If you look at women over 30 years of age, sensitivity is pretty good, specificity is not great, and you're still referring 23% of your population for colposcopy and biopsy, again, very expensive. Okay, so what about the idea that you're going to screen with HPV and come back with cytology? So anyone who's positive for high-risk HPV, you'll then do a cytology on and only biopsy those. Well, the problem is your sensitivity goes down. Because you catch a lot of them with the HPV screen, but then we know that cytology is insensitive, and so your specificity is high and your sensitivity is intermediate. Now, this actually may not be so bad if you think about it for a second, because we know that cytology is picking up the changes for the most part that are HPV related. And we've seen that many of the high-risk changes might go away in these young people. So maybe after 2 rounds of screening, this might not be such a bad approach. There are huge trials. The study we did was about 5,000 women. There are huge trials now going on in Europe that are going to answer these questions. This one may work, I'm not sure. But if you ask me, as I said here, the problem with this one is detection of HPV this is another thing. So before we move on to the last one, I just want to tell you that we've been talking about screening with HPV. There are a bunch of articles, there are a bunch of people who have suggested that you might want to screen with other markers that are related to HPV, like P16, markers of proliferation. And these are really of no interest at all because they're simply surrogate markers of HPV, so they don't do as well as HPV. Okay, so lastly, in summary, HPV primary screening is good sensitivity but poor specificity, and cytology-based primary screening is good specificity but poor sensitivity. Okay, so I think that if you ask me, the only logical approach would be to screen with HPV and then to decide among the people who are positive for HPV, who really has developed changes that we know are associated with cancer, that they're on the road to cancer. Most people with HPV infection we know are going to get well, and it's no big deal. They're going to have the infection for a year or two, and that's it. But some women are moving down that pathway to cancer. So what we need is biomarkers that tell us, that indicate that the presence of carcinogenesis, not of HPV infection. And the referral, we should refer those people to biopsy and look at their molecular profile over time, and those are the ones who need definitive treatment. So I'm not going to talk about this today, and I've left lots of time for questions, but one of the things we've been looking at is the use of detection of aberrant methylation. And this just shows you, we used a candidate gene approach. That just means that we went to the literature and we picked out genes that were aberrantly methylated in other cancers to see what it would look like if you use this to screen people who, for cervical cancer, for CIN3. And as you can see, the first line is cytology, which we look at cytology, and this is biopsy-proven CIN2 to 3, biopsy-proven CIS. We can pretty well do as well as cytology with the presence of 3 aberrantly methylated genes. Since this study, we've done what's called genome-wide searches we and others have done this, and to look at a panel of markers that would be more sensitive for detection of people who are really at risk of cancer. Now, one of the problems is that here the gold standard is biopsy-proven CIN2 to 3 or CIS. And as I've told you, we know that only 30% of the people with CIS are actually on the road to cancer. So one of the problems is what is your gold standard when you develop these assays? Okay, so my last slide here is the other problem that I alluded to. So clearly we're going to have to change our screening approaches. But the problem's going to be bigger than I told you, because we have been talking about about screening with HPV. But we know that the positive predictive value of HPV screening and that of cytology for CIN2 to 3 will decrease as an increasing proportion of the population becomes vaccinated against those types of high-risk HPVs that account for most of the high grade, right? Because the positive predictive value varies with the prevalence of what you're looking for in the population, and we're going to eliminate a lot of those things by the vaccine. So we're going to have more and more and more and more false positives. So we really got to stay away from HPV and cytology in screening, and we're going to have to develop these markers that will allow us to differentiate between those people who are just infected with HPV and who really at risk of cancer. So conclusions. Okay, so as I was talking to before we began, 30 years ago, believe it or not, this shows how old I am, when I finished my training, we did not know that HPV was the cause of cervical cancer. We had just begun to think about it. And in the last 30 years, laboratory studies and epidemiology studies, including our own, have defined the relationship between high-risk HPV infection, cervical neoplasia, and cervical cancer. And this, as you can see, is rapidly changing our approach to cervical cancer control, and it's going to continue to change our approach to cervical cancer control in the future. Moving the things that we have found in the research lab to the clinical setting is somewhat confusing, and that's why we have all these contradictory recommendations coming out. But in the end, I think it's it's going to be exciting and make cervical cancer control more cost effective. And the last thing is that it's going to allow us, the cervical cancer is a great model for carcinogenesis. For example, I can't think of another system where we could look at the natural history and predictive value of, for example, histone modifications and epigenetic changes, because you can sample over time and see what happens. So I think that although we've essentially cured the or solved the cervical cancer problem by development of a vaccine, we're going to be learning a lot more in the future about cancer in general by looking at these new markers in the evolution of cervical neoplasia. Okay, so questions.
So, do you think that we'll ever be able to culture?
You have to scream. I can't.
Do you think we'll ever be able to culture HPV, and why or why not?
I don't think it's really important to culture HPV. I mean, there are some systems for culturing HPV 11, but really that's not what you want to know about it. What you want to know is besides the typing, there are a bunch of studies which I haven't talked about looking, for example, at what causes persistent infection. That might be related, for example, to methylation of various portions of the genome itself, of the HPV genome. So I think there's lots of interesting things that can be done, but I don't think culture, that's not a high priority in trying to figure out, understand the HPV infection.